Hello everyone, my name is Ankur and welcome to RJPT Simlab simulation. In this video, we will study about the potential eye irritation or corrosion caused by a test substance using albino rats. At the end of this video, we will be able to tell about the effect of a test drug on the eye irritation or corrosion which is caused by the test drug on albino rat's eye. Now, let us start with the experiment. At first, let us see the procedural part. The first step in the procedure says about the animal selection. We have to use healthy albino rabbits. Then the second step says the test substance application. We have to administer a single dose of test substance into the conjunctival sac of one eye of each rabbit, which means we have to administer the single dose of the test drug into the conjunctival sac of one eye of the each rabbit. Then the other eye serves as a control and remains untreated, which means on the one eye of the rabbit, we have to introduce our test drug, and on the second eye serves as a control. We have to name it as a control eye, control part, which remains untreated, in which we will do not administer any drug. The third step says about the observation and scoring. It says observe the treated eye at specific intervals 1 hour, 24 hours, 48 hours, and 72 hours, which means that we have to observe the eye after the drug administration in 24 hours, 2 days, and then 3 days, 72 hours. Then we have to record signs of irritation or corrosion, including ulceration, corneal opacity, iris inflammation, and conjunctival inflammation. These are all the effects, which means these are all the effects that we have to observe in the rabbit's eye after the drug administration during these intervals, time intervals. Then we have to score the severity of each effect using standardized scale, example, Gray's scoring system, that we have to variate between the effect or these irritations caused on the rabbit's eye after in specific intervals of time, and we have to score the severity of how much the effect is severe by using a standardized scale. We can use Gray's scoring system. Now let us move on to the fourth part, the result and interpretation. Based on the observations and scores, we have to categorize into three parts: non-irritant, irritant, and corrosive. The non-irritant is the part where if no significant effects are observed, which means after the after the drug administration there are no significant effects are observed, then it says that the drug administered test drug is non-irritant. It says irritant if mild to moderate effects occur, which means when we observe mild or any moderate effects on the rabbit's eye on which we have administered the drug, then our test drug can be said as irritant. Then, if the severe effects are evident, which means when we observe the effects, there are effects are very severe. It has shown a great effect on the rabbit's eye. Then the test drug can be said as corrosive. Now let us see the observation part. The observation says of four categories. Ulceration. These are the effects that we have observed. The first one says ulceration. Ulceration refers to the formation of open sores or wounds on the eye surface. It can be a severe sign of eye irritation or corrosion, which means ulceration is the formation of open sores in the eye or wounds or the surface of the eye, and it is a severe sign of eye irritation or corrosion. Then opacity of cornea. Opacity of cornea indicates cloudiness or loss of transparency in the cornea. Cornea is a part of the eye over which a cloudiness appears, which means we cannot see properly. On the layer of the cornea, on the surface of the cornea, a cloud-like shape structure is formed, which disrupts the vision, which covers the front part of the eye, and it can impair vision and is often associated with eye damage. Then the third effect is the inflammation of iris. Inflammation of the iris involves redness, pain, and sensitivity to light, which means on the inflammation of iris, we observe redness in the eye, pain in the eye, and sensitivity to light in the eye. It affects the colored part of the eye and can be indicative of eye irritation. It indicates as an eye irritation. Then the fourth effect is the inflammation of conjunctiva. Conjunctivitis, it is a disease of the inflammation of the conjunctiva, a part of the eye, results in redness, discharge, and discomfort. It is a common response to eye irritants. This is these are the four different effects which we will observe while performing the experiment. Now, let us perform the experiment in the RJPT simulatory laboratory. As you can see, this is the UI of the RJPT Simlab, and on the left side we have a toolbar over which our rabbit is placed, and on the right side of the toolbar we can see a rabbit holder and a dropper. The rabbit holder will hold the different rabbits, and the dropper will help us to administer the test drug into the eye of the rabbit. As we can see here, we have six different rabbits, and we will administer test drug on these six different rabbits, on the eye of these rabbits. Now we will apply the test drug on either eye of the rabbit, on the left eye, or we can apply on the right eye, only in the one eye of one rabbit. So now we will administer our test drug with the help of the dropper. As we can see here, we have administered our drug solution. As we can observe from the left side of us, we have administered the drug on the right eye of each rabbit. The test drug is being applied on the right eye. So now, after administering the test drug, we will inspect both the eyes after 24 hours of drug administration. So now 24 hours has been passed. So now we will observe the effect of the test drug on these six different rabbits. As we can see here, on the first rabbit, we have observed the opacity of the eye cornea, which means cloudiness has been appeared on the cornea of the rabbit. On the second rabbit, we have observed ulceration, which means opening of the sores in the eye surface of the eye, ulceration. Now we'll see the third rabbit. The third rabbit has shown conjunctiva. It has shown conjunctivitis. And on the fourth rabbit, we can observe the opacity of the cornea. The fourth rabbit has shown opacity in the cornea, which means the cloudiness has appeared on the surface of the cornea. Then on the fifth rabbit, we can observe conjunctiva. It has also, we can by observing the redness in the eye. And then on the sixth rabbit, we can observe the inflammation of the iris by changing in the different colors. It has shown the inflammation of the iris. Now we will observe the control eye, the eye over which we have not administered any drug. As we can see here, the eyes of all the six different rabbits are normal and has shown no effect as in comparison to the eye in which we have administered the drug. As we can see, the eye appears very normal. So now we will inspect both the eyes again after 48 hours of the drug administration, which means after 2 days, after the completion of the 48 hour. As we can observe on the first rabbit, again we have observed the opacity of the cornea. As we have earlier observed on the same rabbit, that it has shown opacity of the cornea. Then on the second rabbit, we have observed ulceration, as we have already observed ulceration of this rabbit. Ulceration is the opening of the sores or wounds in the surface of the eye. Then the third rabbit has shown conjunctiva, is the conjunctivitis, which is the redness of the eye or irritation. Then the fourth rabbit has shown opacity of the cornea, the cloudiness appearance on the surface of the cornea. And the fifth rabbit has shown the conjunctivitis. As you can see, according to the redness of the eye, this is conjunctivitis. And then on the sixth rabbit, we can see the inflammation of the iris. With the observation of the different colors, we can see the iris has been inflamed after the 48 hours. And on the control eye, over which we have not placed or administered any drug, we can observe that no effect has been observed. As we can see, all the six rabbits have the same and similar eye as before. No effect of the drug has been passed on to this eye. It seems very normal in state. By clicking the observation table button, we can see the observation table. As we can see, we have the two categories, control eye and the test eye, and the response recorded in the day 1, day 2, and then further we will record the response in the day 3 also. And then we will clear the result of the 3 days collectively response. The day 1 has control eye has shown no reaction, and the test eye has shown ulceration, opacity of the cornea, inflammation of iris, inflammation, and conjunctiva. And the second day also, the same rabbits have shown the same effect: ulceration, opacity of cornea, inflammation of iris, and inflammation of conjunctiva. Now we will move on to the third day, which means we'll now inspect both the eyes again after 72 hours of the drug administration. As we can see here, again, the first rabbit has shown opacity of the cornea, which is the cloudiness on the surface of the cornea. Then the second rabbit has again shown the ulceration. As we can see here, ulceration is the opening of the wound or the sores on the surface of the eye. Then the third rabbit has shown conjunctiva, as we can see with the redness of the eye. And then the fourth rabbit has shown opacity of the cornea. As we can see here, the white is the cloudiness part. And then the fifth rabbit has shown conjunctiva. As we can see here, the redness of the eye shows conjunctivitis. And then at the sixth rabbit has shown inflammation of the iris. As we can see here, the different colors shows the inflammation of the iris. And now we will move on to our control eye. Control eye seems again normal in condition. All the six rabbits seems the eye of the control eye as normal. As we can see here, it seems very normal and pinkish in color, which is in very normal state. So now we observed all the rabbits' eyes condition on day 1, day 2, and day 3. And collectively, we can say that the control eye has non-toxic drug, which means we have observed no reaction, and which says it is non-toxic, which means we have not administered any drug. And then the test eye shows the effects that we have studied: ulceration, opacity of cornea, inflammation of iris, and conjunctivitis, which means the result says that it is toxic, that the test drug which we have used is toxic. So now we have completed our experiment. I hope that you have learned about the potential eye irritation or corrosion which is caused by a test drug on the albino rabbit's eye. As we can see here, this is the written portion or manual portion of the experiment. We can read it and go through the experiment before performing the experiment into a simulatory laboratory. So now we have completed our experiment, and now we have come to the end of this video. Thank you guys for watching this video, and for more such learning content, subscribe to our YouTube channel RJPT Informatics and follow us on our LinkedIn profiles, Instagram, as well as in Facebook. We update and upload videos of our learning content over these platforms. Thank you.